So in the last video we were talking about what ChIPnCHOP did, how did it work, what inputs did it need, and which outputs did it give. Now we're going to explain a way, we're going to guide you through an example that is provided into the test folder in the repository. So for this purpose you will need to firstly download the directory and install it. Just really simple, you just go to ChIPnCHOP and go to this green button that's saying code. You just copy this URL and you git clone the link. It must not take long, and here you go. What this ChIPnCHOP repository has, it's really simple. You have the README, the license, a format that is needed for the README, a parameter file that is crucial for this whole pipeline to work, and is the main troubleshooting issue making. And then you have the core of this pipeline. This is the scripts folder that has a ChIPnCHOP script and other scripts like sample processing or target gains. We're not going to get through this, but they're open and available if you wish to collaborate in some way. Then you have the test file in which you can find transcription factor example. For instance, this test dataset contains this chromosome FASTA and this chromosome annotation. Those just are from chromosome 1, but they are really useful for the purpose of this example. Apart from that, you have the ChIPsec datasets and the input of that ChIPsec analysis. So we're creating a folder that is called, for example, datasets, and these datasets may contain all the information necessary to carry on the analysis. So we're copying every file that is contained into test dataset now. So we're going to copy ChIPnCHOP test, transcription factor, test dataset, and all that is included through the datasets folder. Apart from that, we're going to copy the essential parameter file. ChIPnCHOP parameter file to datasets 2. We're moving to datasets now, and we're modifying the parameter file. Okay, it looks good. We copy the present working directory, right? And we go through the parameter file. Which is the working directory? I will set the working directory to the test file that I created for this particular example. I will name it, I don't know, dataset, I don't know, PRR5 YouTube example, and the genome directory is going to be called this. Yes, we have the chromosome 1 here. We also have the annotation in the same folder. We have just one chip sample, one input sample, and the chip, both the chip and input samples are contained into the same folder. So I don't know if they are named the same that is provided here in the parameter file. I will say they are, but I'm not really sure about this. We're going to check that out now. and the installation directory is this, ChIPnCHOP. Perfect. So input PRR5 chromosome 1 FASTQ. Let's see. Input PRR5 chromosome 1 FASTQ. It's perfect. Now it looks good. Let's see if it is. So we go back. We bash the ChIPnCHOP scripts, ChIPnCHOP, and we specify, and this is really important, to specify the whole path to the parameter file. So we go home, go to math, YouTube, test YouTube, datasets, and parameter file. There are two options. If I was working with a transcription factor that does not span a lot in the way that large regions of DNA, rather it does take small portions of DNA, we would write this transcription factor. If I was working with a histone or some epigenetic marks, I would prefer to use the HI parameter that is going to assume that you're working with some sort of protein or protein modification that is spanning a large portion of DNA. So I already ran this pipeline, and now what it is doing, it is analyzing, it is indexing the genome, right? As it is not a large genome, it might not take long, but keep in mind that the genomes that you are working with, like the ensemble that I showed you, might take quite long. A few minutes to might be the normal thing to expect. So now it is doing the FastQC, and now I bet it is mapping the reads to the index genome. You may need to wait quite a lot in the case of normal ChIP-seq samples, but in this case it won't take that long, just a bit. You see, as this is an example, the alignment is going to be really good, like 97.84%. It is not really the usual things to see, but if it does, it is perfect. There's some information. The 99.96% of overall alignment rate is really good. So it has also mapped the input files, and now it has done the max to peak. It is doing it now. The max to code peak. Once it has been done, it starts to run the R script that is going to annotate the peaks to the function, that is biological processes, molecular processes, cellular location. Okay, now it's annotating by ChIP-seq. There are going to be some files that are generated that we need to check out to see if they are correctly estimated. So 584 genes have been determined to be a target for PRR5 for this example, and those are going to be stored in a txt file that we're able to see afterwards. You may want to change a few parameters. If so, just go through the results, and there will be a transcription factor, this or histone R script, in which you may be able to change and adjust the parameters at your will. Mm, Afterwards, you will have this gene ontology enrichment analysis, and Homer analysis is taking place. It will set a few background sequences from the TER10 genome that it has stored. It is important to install it, but if you have previously used Homer, there might be no issues, but and that's pretty almost everything. It will take a while because it needs to find this enrichment of all the motifs that it may find, but that's pretty all to see from now. If you would like to, I will cut this now, wait till the results are, and make a copy to my personal, to my local directories to show you how the results are stored. It is quite intuitive, but if you're not used to see it, you may find quite lost while searching for them. So, see you in a few secs. Welcome back. Well, after finishing this analysis, it says really discreetly that it has been done. But once you have safe copied the whole directory, I copied the annotation, genome, results, and samples. You can find them at your desired folder. For example, I have them at peer YT example. In this case, I have them at results, and here in the results file you may find different things. First of all, the motif finding, that is the last thing you have analyzed, might be useful to identify some interesting de novo motif finding, for example CACGT, or in the case of, you see, the CACGT that is targeted by ABF1, for example, the BCIP. There are many others motif finding that can be recognized by PRR5. It might be useful, and you can analyze pretty almost everything. For more information, you can just go to the documentation of Homer, and they have explained with quite good documentation everything. Then you may find the regular enriched TXTs and the R plots. In this case, as we only have one chromosome, you will see only just one line, but there is everywhere that PRR5 has explored and has bind. So you see this, that this promoter spans 65% of all the annotations that it has done, and that it is contained within 1 kilobase. The transcription stats are quite close, and a similar result is observed by this plot, although this is not really clear because this is just a toy example. Then you got these annotations, the response to hypoxia, to oxygen levels, and the p-adjust value. The same goes with this dot plot and a CNET plot. We can see it by a network So there's pretty much everything that this can be done. But never forget, you can actually get into this script and modify pretty much everything, obtain much more information, use your own packages that can be useful for analyzing the peaks. Always remember that this reads the peaks from max2 analysis, but that's pretty much everything. I could say that's everything that ChIPnCHOP is able to give to the community, and we're happy with it. But any suggestion, you can just leave it at our Discord homepage. Of course, you can just go by GitHub and write it through the forum, or you could go via the chat and Discord and just write us. Just log in and be free. Just feel free to suggest any other suggestion that you may have. Okay, so that's pretty much everything. We're happy to have you watch everything till now, and we hope you'll see you again. Goodbye.